Hello, my name is Katerina Jordan, and I'm a researcher in the Department of Plant Agriculture at the University of Guelph. And today I'm here with my two master's students, Kieran Maloney and F.A. Elgin, and we're here to talk to you about take-all patch, a rather important root disease of turfgrass. So my fascination with take-all patch started a little over 10 years ago when we would receive samples in the GTI diagnostic clinic. So I would notice these patch diseases that would come in on bent grass while the annual bluegrass was left asymptomatic. And I didn't notice any evidence of foliar mycelia or any evidence of diseases on the leaf tissue, but I did notice these dark threads on the root tissue, known as runner hyphae. And so I would diagnose this as take-all patch. And so I found that the prevalence was usually greater in from provinces like Alberta and Saskatchewan and Manitoba, as well as much of Ontario. And the other thing that was really interesting was that we would be getting samples from courses that were 10, 15, sometimes 20 years old. And they would be rather surprised that they were being diagnosed with take-all patch. And so part of the reason that this was curious is that take-all patch is known to actually disappear after about 5 years. It usually tends to appear on newer or newly renovated golf course greens or fairways or shortcut bent grass turf. But we do tend to see it disappearing after about 5 years in a phenomenon known as take-all decline. And so the theory is that we have antagonistic microorganisms within the soil that will outcompete the causal fungus. And so over time, it then tends to decline. And we noticed that we were seeing this disease on golf courses that were much older than 5 years of age. And so I started putting together some ideas, and we know it to be a generally high pH disease or a disease that occurs in alkaline soils. So I started looking into it and thought we might want to look at what's going on beneath the soil in these alkaline soils. As well, we also saw that there would be varying symptoms. And when a DNA test was done at lab services at the University of Guelph, they weren't finding evidence of the causal agent, at the time, Gammanomyces gramineaceae. So I also surmised that perhaps we had a second pathogen at play here. And over the years in my laboratory, we have actually confirmed a second pathogen of take-all patch known as sloeimyces cylindosporus. So I started reaching out to the Ontario Turfgrass Research Foundation and suggesting that this is a pretty serious disease and that a lot more golf courses are suffering from this than they might be aware of. And that it was worthwhile to investigate both this second pathogen, which we really knew so little about, but also to investigate what's going on beneath the soil, and particularly the living part, to look at some of the properties of the soils that tend to lead to a buildup of these antagonistic microorganisms and why we're not getting take-all decline in certain areas. So OTRF has very generously funded two projects now to look at this. And so with that, I'm going to introduce the two graduate students working on this, and they'll tell you a little bit about their interest in the study and what they hope to achieve.
Yeah, so hi, I'm Kieran Maloney. I'm a first year master's student. I just finished my first year. And I'm one of Katerina's graduate students working on this take-all patch project. I kind of got interested into this project when I first met Katerina. She had kind of mentioned this project to me, and it kind of sparked my interest because as a golfer myself, I love being out on the course. I love playing golf. But the one thing that maybe I would like to see different in golf courses is a more sustainable approach to treating diseases such as take-all patch. So my goal for my project is to first determine the presence of both Gammanomyces avinae and sloeimyces cylindosporus within golf courses in Canada. As Dr. Jordan has just said, these are two very different pathogens that cause the same disease. And once I determine the presence of each of those pathogens, I want to determine what are the conditions in which both of those pathogens are found. Are they found in very different conditions or very similar conditions? And those conditions include some soil properties, so soil pH, soil fertility, thatch levels, as well as some management practices. So what is the irrigation regime, the fertilizer regime, how often is aeration occurring on the course? and as well as the environmental conditions of the course. So that's your air temperature, your soil temperature, your rainfall, how much shade or sun is the golf course green that has take-all patch on it, how much is it getting within a certain day, month, or even growing year. In the past, researchers have looked at trying to lower the pH of the soil in order to treat this disease without using fungicides. So last summer, as a undergraduate student in Dr. Jordan's lab, I set out a field trial in which I tried to lower the pH of our alkaline soils here at the Guelph Turfgrass Institute in order to decrease the incidence and the severity of the disease itself. So I was able to decrease the soil pH from when I started in May to when I finished the trial in October. However, I did not see any variable effects on the presence or the severity of both pathogens. This is a trial I hope to continue into the future in my master's program and hopefully tweak a couple things in order to maybe see some variability like that I was hoping to see. So as of right now, the use of preventative fungicides is the go-to way to treat this disease on courses. Um, it is known that this could lead to resistance in the pathogens themselves against the fungicides, but the use of these fungicides also alters the microbial community of the golf course soils. And ultimately, I want to find a way to treat this disease without affecting these communities in order to have a more sustainable approach to treating this disease.
Hello, my name is Efe Engin, and I recently just graduated from my Bachelor's of Science in Agriculture here at Guelph. I've joined Dr. Jordan's lab as a research assistant to learn more about the project and get an understanding of the methods, and I'll be joining as a master's student in the fall. Originally, why I got interested in this project is when I took the biocontrol class taught by Professor Jordan. And I really wanted to understand how microbial communities work, how they can help fight against disease. And I want to investigate this further with the take-all patch. So what I want to accomplish with this project, I hope to get a better understanding of the growth and survival of sloeimyces, cylindosporus, and Gamenomyces avinae in a variety of conditions such as temperature, pH, and also against microbial communities that we know to fight against these pathogens, such as the Pseudomonas. I also want to test the efficacy of a biocontrol against the pathogen, as I think it can be really helpful to golf courses to reduce their chemical controls and help increase more sustainability. And finally, I would like to test the susceptibility of various cultivars of creeping bentgrass against both these pathogens, and this will help make better recommendations to golf courses on what they can do.
So with the help of both Kieran and Efe, we hope that we can have an answer for golf course superintendents who have been struggling with this disease for a really long time. And as Kieran said, the number one way of preventing or dealing with this disease is generally the application of fungicides, which can be relatively effective, but they do require very specific timing, and they do need to be watered in to actually manage the root system. So it can be a bit of hit or miss sometimes. So finding alternatives, I think, will be really helpful. So a special thank you to the Ontario Turfgrass Research Foundation for investing in this project and for supporting Kieran and Efe. And hopefully we'll have some answers for you guys at the end of the project. Thank you so much.